Hello everyone. First of all, I'd like to thank the PGO conference team. I really appreciate the help and the invitation. I'm Yuanyuan Chen, a postdoc researcher from Huazhong Agricultural University. It's a great honor to present our recent research about a heavy metal transporter HMA3a in maize. The rapid urbanization and the industrialization have caused the widespread heavy metal pollutions in agricultural soil, which pose serious threats to food safety and public health. Among the deleterious heavy metal, cadmium is the most bioavailable toxic pollutant and easily transferable through the food chain. While several physical and chemical strategies have been applied to immobilize cadmium in the crop lines, it is promising to cultivate field crops that produce enough seafoods for human consumption and simultaneously remove the pollutants from contaminated arable lands. For this purpose, it is important to understand the mechanism of cadmium accumulation in plants. In recent years, a lot of studies have demonstrated the major process involving cadmium root uptake, root-to-shoot translocation, and accumulation in Arabidopsis and rice, but few genes have been functionally characterized in maize. In this study, we performed a genome-wide association study for maize leaf cadmium content in a synthetic population, cubic, and a diverse maize association mapping panel, AMP. We detected a major peak on chromosome 2 that highly associated with the leaf cadmium content in cubic population. And according to the parent IBD status of these locus, the cubic offspring were classified into 16 groups, and the G53 group greatly exceeded the China guidance levels of cadmium contamination in cereals. We also identified the same QTL in the association mapping panel, and there are two genes at this region were annotated as heavy metal ATPase, which are homologous to HMA3 in rice. So we named them as HMA3a and HMA3b. RNA-seq analysis revealed that HMA3a was relatively high expressed in the roots, while HMA3b was essentially not expressed. To investigate whether HMA3b is functional or not, we obtained two independent knockout mutants and two overexpression lines. It was noticeable that the leaf cadmium content of HMA3b knockout mutants was not significantly different from the transgenic receptor KN5585. However, overexpressing HMA3b did not alter cadmium accumulation compared to the non-transgenic lines. So, taken together with the expression evidence, it is demonstrated that HMA3b was not involved in the regulation of the cadmium level in maize. The significant associated variations identified on HMA3a in the association mapping panels can classify all the 409 maize lines into 6 haplotype groups. Comparatively, the leaf cadmium content of HAP6 was not significantly higher than the other 5 groups. Moreover, the leaf cadmium content of maize lines carrying in 381 was significantly higher than that of the lines not carrying this in 381 in the association mapping panel. So we found that the INS 381 is annotated as an specified class LTR retro transposon. Subsequently, we resequenced the HMA3a in the high cadmium accessions and low cadmium accessions, and verified a big polymorphic insertion existed in the first intron of HMA3a, which is just the INS 381. By sequencing the full coding sequence of HMA3a, we found that HMA3a in all high cadmium accessions like G53, MOL17, and KM5585 is mistranscript, which generate a premature protein termination. In addition, the progenies in cubic harboring this intronic insertion accumulate more cadmium in the leaf and kernels than the lines without this insertion, but the expression level of this gene in the roots were not statistically different among these individuals. So the above evidence implies that this LTR retrotransposon is a causal variant that did not alter the expression, but induced the dysfunctional protein. Therefore, we speculated HMA3a might be the causal locus of nature variation in the leaf cadmium of maize plants. In support of this, knockout lines of HMA3a did not result in any change in cadmium concentration either in the leaves and kernels. By contrast, compared to the wild accession B73, two EMS mutants accumulate much more cadmium in leaves and kernels. To investigate the molecular function of HMA3a, we transiently expressed the gene filled with GFP in a Rhabdopsis mesophyll protoplast and found that the fusion protein was located at the tonoplast. We also grow the OE plants and the CK plants in the cadmium-contaminated soil and extract the protoplast from the root tips in both lines. It is observed that the flowering signals from the OE8 protoplast were stronger in the vacuum than that in the cytoplasms, while the signals from the CK protoplast were weaker in the vacuums than that in the cytoplasm. So this data confirms that HMA3a is a core valve located at the tonoplast which sequesters the cadmium into the vacuums in the root tissues. So this result indicated that increasing the expression of hyperfunctional HMA3a is of great value for improvement in cadmium tolerance in maize. So we carried out the field trials with overexpression lines and the non-transgenic lines under 3 cadmium levels, such as background is 0.1 ppm, slightly polluted is 1 ppm, and heavily polluted is 3 ppm. Across the 3 different cadmium level trials, the yield development of CK plants or remarkably impeded in the 3 ppm field. And the kernel weights of the OE plants and the CK plants were not significantly different in the background and the lightly polluted soil, but the kernel weight of the CK plants were tremendously decreased in the heavily polluted soil. All of this data indicated that heavily polluted soil can significantly affect the normal growth development and the yield of the maize. By contrast, HMA3a overexpression lines can produce normal yields in all the 3 field trials, and the yields were not significantly affected even in the heavily polluted field trials. It is noticeable that the cadmium content of OE kernels were much lower than the national edible agricultural product quality safe standard levels in China. To assess the ultimate potential of HMA3a in improving cadmium tolerance and food safety, we grew these lines in pots containing field soil supplemented with 18 ppm cadmium and without cadmium supplementary. So the transgenic receptor KN5585 and CK plants performed well in cadmium-free soil pots, but they were severely impaired either in the growth or yield production when exposed to an extreme cadmium-polluted environments. By contrast, the OE plants were not affected for the growth and only mildly impacted with regard to the yield when exposed to cadmium contamination. The cadmium levels of the OE kernels were still far below the food safety standard. All the evidence collectively provide broad perspectives for the application of alleles of HMA3a in resolving the cadmium pollution problem in the agriculture. So brief summarize what we did. We performed the GWAS and characterized an ATPase transporter HMA3a responsible for the cadmium alteration in maize leaf. Moreover, we identified an LTR transposer inserted in the first intron of HMA3a result in the premature translation termination in the high cadmium maize accessions. And finally, we determined that overexpression of HMA3a produce cadmium-free maize kernels without corresponding yield and quality penalties in the heavily contaminated soils. So our results provide the broad perspectives for the application of the alleles of HMA3a in resolving the cadmium pollutions in agricultural soils. At last, many thanks to my lab and our collaborators, Dr. Chao Daiying and Dr. Chao Zhenfei from Chinese Academy of Science, and this study was supported by the National Science and Foundation Center. Thank you for your attention. I'm happy to take any questions.